Well, thank you for the introduction and for the opportunity. Oh, sorry. The red arrow goes back here. And the opportunity to present to you our TRACE program on the qualification of patient-derived biomimetic liver MPS as drug development tools. So metabolic dysfunction-associated steatotic liver disease is a mouthful, but MASLD is a progressive, complex, heterogeneous disease that impacts a global population of over 25%. Now, the pathogenesis of MASLD is complex due to the various patient heterogeneity, including genetics, lifestyle, environment, and other comorbidities. And over the past 10 years or so, there's been a lot of research and development for getting therapeutics for MASLD, but there's only one FDA-approved drug. Yet, only a minority of patients respond to this drug. So MASLD is an example of liver diseases, which in general, liver diseases are very challenging for drug development. And one of the challenges for drug development that liver diseases pose is estimating dosing for the intended population. Clinical trials generally start with healthy volunteers to determine what the dose ranges are going to be. However, in liver disease, the drug bioavailability can be altered. It can be increased or it can be decreased. And one of the mechanisms by which that happens is because obviously the liver is a main organ for clearance of drugs, and that is what affects the blood levels. And so you can see that liver diseases can actually change the metabolic profiles of drugs. It can increase the clearance or decrease the clearance, which changes the Cmax. And that also is true for MASLD. So the unmet need that we see is that understanding how to effectively translate pharmacokinetic data from healthy volunteers to the intended patient population with liver disease to inform decisions of dosing for phase 1 as well as the phase 1b/2 transition. Also along these lines, if it goes, yes, is that liver diseases, including MASLD, can alter the sensitivity of the liver to hepatotoxins. So another unmet need in the area is understanding how to effectively translate the safety data from the healthy volunteers to the intended patient population. And finally, a major challenge in drug development for not only liver disease, but other diseases is identifying the efficacious drug candidates and the responsive cohorts. You can see here that a number of drugs have been tried for MASLD. 96% of them, which we have clinical data on, have failed. And the one drug, resmetirom, that has been approved, you can see on the right, only is responsive, only 30% or so of the patients are responsive. And so effectively identifying the right drug candidate and the right patient cohorts to advance to clinical trials is an unmet need. So to address these challenges, we've been developing our LAMPs, or liver acinus microphysiology system, and our VLAMPs, which are structured biomimetic liver MPS, as drug development tools. So the LAMPs on the left is constructed from using the 4 major liver cell types, which we construct in a single chamber device, which allows us to control flow rate of media to be able to set up different metabolic zones. For example, zone 1 and zone 3, the liver acinus, and we can study the disease and the effects of drugs on the disease in those zones independently. The vascularized acinus liver system, the VLAMPs, is in a 2-chamber device with the same cell types, but this allows for a continuous zonation created in one device to maximally recapitulate the liver acinus. But this also allows for optimal communication between the vascular and hepatic chambers for example, including immune cell filtration or drug delivery or circulating factors in a more biomimetic fashion. Now, I'd like to point out that what we're doing is that we actually are qualifying the biomimetic part, which is the biology of the model, independent of the individual device. And we are testing a number of different devices for these models to avoid any potential supply issues down the road. But our overall goal is to qualify these drug development tools for 4 contexts of use. The first two contexts of use use the LAMPS model. The first context is for establishing the hepatic clearance of drug candidates in patients with late-stage MASLD to provide supporting data to determine drug candidate dosing in MASLD clinical trials. The second context of use for the LAMPS is to establish hepatic toxicity of drug candidates in the patient population with MASLD to provide data to show acceptable toxicity in the human disease state for inclusion in the MASLD trials, also to aid in the determination of candidate dosing or dose limits in that population. The second two contexts of use, or last two contexts of use the VLAMPS model. The first one is to provide data on the anticipated efficacy of drug candidates in the human disease to support advancement to a clinical trial. And the fourth one down there is using the VLAMPS to predict the responsiveness of individual patients to drug candidates for their inclusion in clinical trials. So there'll be the clinical trial cohort selection. We're working on all four of these at the same time, but we're qualifying them one at a time, and we're starting with the clearance context of use. And so this is just to show that we have the protocols in place for the LAMPS model for predicting clearance and hepatotoxicity. We measure a number of mechanistic measurements. We collect a number of phenotypic and functional measurements for the model to show that the model is it's a functional liver and we can induce the disease. We've demonstrated the reproducibility of the LAMPS model, both internally as well as across labs. And we've also demonstrated that the MASLD phenotypes are reproducibly induced in the LAMPS. What we use is we change the medium content. We have what we call a normal fasting medium to induce a healthy liver, or we have an early metabolic or EMS medium, which induces a MASLD disease state. And you can see that looking at three different metrics or phenotypes for MASLD, we can see an increase in steatosis or stellate activation or in prefibrosis with collagen 1A1 when we go from a normal to a diseased medium. And we've demonstrated the reproducibility across multiple studies here, as well as across multiple patient cell lot of cells. We've also demonstrated that the LAMPS is metabolically active. We use a series of probes to look for the disappearance of the parent compound. We look for the appearance of CYP-specific metabolites, and we've demonstrated how the LAMPS is metabolically capable. And how we put this all together is that we construct the LAMPS using primary hepatic cells collected from normal individuals. We can induce them with either our normal fasting medium to keep a normal healthy volunteer state, quote unquote, or we can induce them to the MASLD state using our EMS medium. And we do assess the hepatocyte function, we assess the disease phenotype, and for the context of use of hepatic clearance, we look at the parent drug levels, look at the disappearance of the drug over time, we look for the metabolite formation. From that information, we can predict the hepatic clearance. We can then go and predict other PK parameters, which can then be used to guide phase 1 dosing or guide dose adjustment decisions in the phase 1B transition. And right now we are working on testing a number of drugs that have altered clearance in liver disease to validate the model. And so this is just an example of how we would apply it in the regulatory decision-making, where we would look at the disappearance of drugs. For example, omeprazole, this is from one of our more recent experiments, where we tested it under the normal condition and under two different disease conditions. We've calculated the clearance, that the clearance is actually reduced in the disease state versus the normal state. And from that, we were able to predict, well, here are the steady state levels if you were given the standard 20 mg per day dose of omeprazole. And you can see that with the EMS and LMS, which are the disease states, that the steady state concentrations are higher than what you see in the normal state. If you need to get down to the normal state for safety reasons, that means that you're going to have to reduce the dose in the disease state from 20 mg per day to maybe 12 or 13, 12 or 14 mg per day. And so, and if you actually look at the package insert for omeprazole, that's actually what's recommended for patients with hepatic impairment to go from a 20 mg per day to a 10 mg per day dose. And so this is how we envision that it will be applied in making those regulatory decisions for the patient population who have MASLD or other hepatic impairments. And so I just want to leave with some questions that we have, either for the panel discussion now, but at least what we want to learn in discussions with the FDA as we move forward is that, is there agreement on this unmet need for being able to have a better way of determining dosing for this intended patient population? How do they see the LAMPS model in filling this need, as well as what are the expectations for further validating the robustness of this model for these needs? And then just finally, just want to acknowledge this a great team of people who have been working on this for a long time in developing the models and helping to push this forward. Thank you.